that's fine. So you can
Yeah,
go
okay.
to the right place. So yeah, floor is
Can I
yours.
take it away? All right. Welcome, everybody. On the back of this envelope, I've written Todd's bio sketch. Todd's a long-time collaborator of mine. I was, in fact, a postdoc in his lab at University of British Columbia, the most beautiful place on earth, for a year. And so Todd's a professor in psychiatry at UBC, and he heads the cognitive neuroscience of schizophrenia lab, the CONUS lab. That'd be right. And he, apart from like doing tons of things because he publishes like literally every 2 minutes, he's developed, and what he'll be talking about today, some really interesting kind of methods for fMRI data analysis. And his particular focus is on identifying brain networks involved in cognition, but also how they are affected by neuropsychiatric conditions. Is that so far sounding correct?
Excellent.
And so we have to be a little bit gentle with Todd because it's 5 a.m. local time. When I sent him the invite, I couldn't work out the time difference correctly and said it was 6 a.m. But turns out it's not, it's 5. So
what's the difference?
A blessed hour of sleep, Todd. It's huge. But Todd also has lives in this house with floor to ceiling windows, and if I ever talk to him on like Skype or Zoom or whatever, he'll sometimes flash the camera around to show me mountains and sea. So let's not feel sorry for him at all. Finally, before I let Todd start, he did say he's happy to be interrupted. So if you want to, I'll sort of monitor the chat, or if you want to raise your hand and I can just point out to Todd someone wants to ask a question, please go right ahead. Otherwise, we'll have the usual questions at the end. Okie dokes. I shall let you, Todd, come back. Oh no, have you disappeared for a reason?
Now I'm gone.
Okay. All right. I will turn my camera off. I'll mute myself and I will let you go.
Okay. Can you see the screen?
Yes.
But not me.
Correct.
Okay. Okay. There's something in the chat already, so just let me know if anything. Just go ahead and interrupt me. Okay. So thank you very much for the kind introduction, Eva, and it's really an honor to address you all today. And I want to talk about task-based fMRI as opposed to resting state, which is much more popular, I guess. So for those of you that don't know, functional magnetic resonance imaging, or fMRI, is a reliable method for measuring to where oxygenated blood is flowing in the brain as a result of neurons firing. So this is very important to keep in mind and easy to forget that we're not actually measuring neural activity, and we may not even be, you could argue that we're not even measuring neural activity, we're measuring something that's related to neural activity. So brain networks detectable by fMRI have been essentially unknown outside of resting states. So you can see my mouse, right? Hopefully, if not, just let me know. So
yeah.
Thank you. So there's two types of experiments, really. There are obviously a lot of different experiments, but generally they can be described as rest or task. So in resting state fMRI, people will sit in the scanner, eyes closed or open, and they'll be told to relax and try to think about nothing in general. So as you can probably imagine, they are thinking some things, but the difference really between a resting state experiment and a task-based experiment is that in a task-based experiment, you know when people, you try to control when people are thinking what. So it doesn't have to be a visual presentation or an auditory presentation, but there has to be a way for people to know at exactly what time they should carry out some type of activity. And there's two general ways to analyze data. One is sort of a network-based type of approach, so that all activity can be split into dimensions of different types of bold signal that's correlated together. So not necessarily neural activity together, but changes in the bold signal over the brain and how those correlate together. And typically, and you can also kind of treat each smallest measurement of brain area called voxels separately, so that they can all be considered a separate dependent variable in a regression equation. And you're trying to predict activity in each region by a model of the bold signal. So typically, resting state experiments are studied with networks, and task-based experiments are studied with each individual region treated separately. But well, when Eve and I started working together, we started developing a method that looked at networks, but not during rest, but during tasks. And it's surprising to me, obviously, since I've been doing this for, well, 15, 20 years, that nobody else has really, there hasn't been a lot of attention on networks involved in tasks. That's rarely studied. And so we've been working on this a long time. So hopefully we'll be able to share some information that will be able to push the field forward a little bit in this respect. And here are the basic questions that would be involved in task state or task-based functional brain networks detectable by fMRI. So what are they? What are their anatomical characterizations? That's the easy part. Well, it wasn't that easy, but it was easier than determining the cognitive functions of the networks. And then also we need to know to what extent these cognitive functions are task-specific. So does every task elicit its own set of networks, or is it that there's a set of networks that spans multiple tasks or even all tasks? And those three questions, if they can be answered well, then we can start to look at how are they affected in brain disorders. And if we have symptoms, my lab studies schizophrenia, and hallucinations and delusions are a particular focus. So is the manifestation of delusions, is that network specific, or does it depend on which task people are carried out? So I'll try to answer some of those questions today. So here's Yoshio Takani, who was a retired professor at McGill University, and Yoshio and I we would spend the summers in Victoria, which is close to Vancouver, and we would talk a lot about multivariate statistics and eat sushi, drink beer. Not a lot of beer, just enough to keep the conversation academic. And we developed this method of analyzing fMRI data. And really, CPCA is not anything to do with fMRI. It's a generalized set of matrix equations which combines multivariate multiple regression and principal component analysis into a unified framework. And first paper on that, well, and then, with talking with Yoshio and reading his papers, I realized that this would be a great way to analyze task-based fMRI. And that methodology was first published in 2006. But that was a previous version of fMRI-CPCA, and the one that we still use now was published in 2011, and Eve and I collaborated on this. And so now it's been 10 years since we published that first paper. And these are the most recent fMRI-CPCA publications. And these we still haven't published the full explanation of the networks that are detectable by task-based fMRI, but they actually are all published in this combination of papers, but still need to put together the full explanation of the methodology with the results. And actually, when even in this 2017 paper, we still didn't actually know the cognitive functions, and we didn't know that the same networks are replicating. So it's only been over the past few years and long days of sort of squinting at computer screens when I finally started to realize that the same patterns are replicating over tasks, and then we started to classify these networks. So I'm quickly going to go over the motivation, and for those of you that are not interested in matrices or methodology, you're completely safe to ignore this slide, and this is for people that just can't stand to look at the results without knowing more details about how it was derived. So this, just orienting you back to this table, and here we have the typically studied networks at rest, and then we have the rarely studied networks during tasks. So the data is set up with the voxels in the columns, and the rows are full scans for TRs. So there's each row is about 2 seconds worth of time in a typical fMRI experiment, and subjects are stacked one on top of the other. So subject 1, subject 2, subject 3 will be stacked one on top of the other. So this data set typically is maybe, if it's 3 by 3 by 3 voxels, there's about 60,000 columns. And the rows, if there are 300 scans per person, 10 people, there might be about 3,000 rows. So this is for about 10 people, it'd be 3,000 rows, 60,000 columns, and this would be a full fMRI data set pre-processed. And so that's why I'm calling this 100% of the bold signal variance. And when this is analyzed with resting state, then you would usually take components out of this data set. And so the thing is that, resting state networks are typically described as, sort of these fundamental networks in the brain that, are not changing. But the thing is, in fMRI-CPCA, what we do is take the timing. So we know the timing of tasks or the thought processes that we're asking people to go through. We know when they're doing that. And we can put in a model that predicts, that is based on the task timing. And when we use that model as an independent variable and the fMRI signal as a dependent variable, we extract about 10% of the variance. So really 90% of bold signal is not related to task timing. But if you extract the 10%, obviously the networks that are involved in rest that would be detectable when 100% of the variants are analyzed, those are not necessarily optimized to explain tasks, because tasks is only 10% of the variance, and that needs to be extracted. When that's extracted and focused on singularly, then you're able to see which networks are specifically related to the task. And they are some, 1 or 2 or 3 maybe overlap with resting state, but most of them don't. So these are the basic, these four sort of principles are the 4 principles that we focused on since the beginning of since we first published the 2011 paper anyway. And these 4 principles make fMRI-CPCA unique. No other technique actually combines all 4, even though many might combine 1 or 2. So first of all, we're not going to be treating each voxel as a separate dependent variable. That would be the typically studied regions approach for tasks. We want to do a multidimensional type of analysis, which will involve, in this case, a PCA. And we want to isolate the task-related variance prior to network extraction, which I just explained. So this blue box is telling us we're looking at this portion of the variance, this 10% that's predictable from a model of the task timing. Multidimensional, focused on the task-related variance. And we're not parcellating the brain into multiple, say like 250 different areas, or choosing any regions of interest as a seed. Every single brain area will be allowed to contribute to the network. So it's a very exploratory type of method. No a priori hypotheses, at least not about individual areas, including all areas. And so that's why this is in green, including all available brain areas. And finally, this is very important, that we don't make any assumptions about the hemodynamic response shape. We actually let the data tell us what the hemodynamic response shape is associated with each network. So we're completely allowed to explore all the brain areas that may be involved and explore what the hemodynamic response shape looks like. And so over 10 or 15 years of doing that, this is how we came up with sort of recognizing the consistencies in the networks that we're retrieving. And so this picture is not new. This is what I just showed you on the last slide, but for those that aren't, that familiar with the hemodynamic response, it's actually a fairly complex shape. So in this is a slide that I created for a study we're doing that tries to find the networks involved in hallucinations. So this would be a person hearing a voice again, hearing a voice again. So they've heard 4 voices during about 15 seconds of time. And the hemodynamic response for the first voice would increase and try to get back to baseline. But then when the second voice is heard, then that would increase again. And sort of you end up with this complex shape because there's usually overlap between the subsequent trials, whether it be a task or hearing a voice. But using finite impulse response models and principal component analysis, this shape can be separated into fundamental shapes that are contributing to the complex shape. And each fundamental shape for the hemodynamic response is associated with its own brain network. So that's sort of all I'll be talking about the methodology today, but we can talk afterwards if people want to get into more detail. So here are the networks, and on the side here I have listed the GitHub address for where you can download these images, and also some more detailed evidence about what the words are that we write in here for what the cognitive function of these networks are. So there's 3 networks that are involved in internal attention, so attention to internal representations. Talk about those in some detail now. 3 that are involved in tending to external representations. 2, auditory processing. These 2 are response networks, so this is a one-handed, two-handed response network. And then this is two types of default mode networks, which are thought to be involved in self-reflection. But typically, when people carry out a task, the default mode networks decrease. And again, here's the GitHub link that you can download the templates and some documents that will provide evidence for the anatomical and functional characteristics of these networks. But again, here are the resting state networks. This is the Yo parcellation of 7 different networks, and they're the names for them. And that again is when you're analyzing 100% of fMRI signal, and I don't think there'd really be much difference between task state and resting state in terms of retrieving these networks, since 90% of the bold signal is not predictable from task timing anyway. But if you look at 10%, take out that 10%, and we use PCA, but you could use ICA, then here are the networks that are carried out. So I hope all that's clear about how important it is to, extract the variance. And this is the whole philosophy of CPCA, that if you can extract variance that is related between two data sets, then the fundamental dimensions of the questionnaire or the data or the bold signal that you're looking at in the first place won't necessarily be optimized to predicting another piece of information, in this case, task timing. And so we can when a new network is retrieved, we can automatically classify it. So this is just new. This is Chantal and Hafsa. We worked together the past couple of years on this. And so now we have a MATLAB program that you can input a new extracted network, and it can be automatically classified into one of the 12 here. But sometimes there are combinations of networks that can get a little tricky. But we don't have this published, so it is available. You have to contact me to sort of, register with UBC as an invention, but it's not made publicly available yet. And what we found is that it doesn't matter which task you use or which scanner you collect the data from, which investigator it is. fMRI signal, when it's task-based, is dominated by these 12 networks. And of course, we're working more on finding sub-networks that activate under certain conditions, but these are the dominant 12 networks. So that's kind of anatomically now. And if you look on the GitHub, we have a whole thing sort of, in detail of every single slice that makes one network different from the other 12. So I obviously don't have time to go through that now, but it's all there for you to see. But so here's the part I want to talk about mostly. What are the function of these networks? So brain networks are more than just a pretty picture. So we're used to seeing the picture, and resting state research has shown us a lot of pictures over the past 15 years. But what evidence is there for the function of those networks? So, unless you have task-based fMRI and you're driving those networks in certain ways, it's difficult to not just speculate about the function, but you have to try to prove that. So that is the domain of psychology. And, for those of us who are trained in psychology, it's sort of, I think, difficult for people to think past just the picture of the brain and more about the cognitive functions. And you get into a domain of reverse inference. So by looking at a particular pattern like this, can we say by that pattern, you might not be able to see the differences between these patterns, from this one picture, but every single signature is completely unique for each network, and it takes some detail and some experience to see that, but it is the case. So since we can find specific patterns, can we associate a cognitive function with each pattern? That is reverse inference, to look at a pattern of brain activity and to know what cognitive process is being carried out from looking at that pattern. So let's see what evidence we have. So this is Nicole Sanford. And she finished her PhD with me, I think, in 2019. And so this is a working memory. So this is a Sternberg type of task that even I worked on, 10 years ago. And it's she is much more of an expert in working memory than I am. But I've stuck with the Sternberg, not because that is particularly interesting from a cognitive psychology perspective, but because it is very interesting from a perspective of reliably extracting networks with fMRI. So in this case, you're remembering 4 letters, or sometimes there's other letters to make it 6. So this is 4 or 6 letters. There's a period of 4 seconds where you're meant to or required to maintain that information. And then after this maintenance period, you've shown 1 letter and just say yes or no whether or not that was presented here. We also have in this experiment conditions where this maintenance period is completely omitted. So it just goes straight from the encoding to the response. And so we have two manipulations, 4 versus 6 letters, maintenance phase versus not. And so, you know, I think what is interesting about reverse inference with fMRI, so, you know, assigning a cognitive operation to an anatomical configuration, is that you end up actually measuring a range of cognitive processes. So notice that if we were to use the Sternberg task for, you know, neuropsychological assessment, we would actually have usually only errors. Reaction times aren't usually the measure of interest. So, you know, we have to, you know, create some manipulations to look at how this changes errors, or, you know, you might have more precise measures of errors, but generally the behavior is fairly unidimensional and not particularly complex, you know, in cognitive psychology, unless, you know, you get into confusion matrices and things like that. But with fMRI, we actually get 4 separable networks in this task, and they're interpretable and they, to some extent, in terms of recognizing the manipulations that we put into this experiment. And the same networks can be retrievable in a variety of other tasks. And so we start to, you know, triangulate what the function of this anatomical configuration is based on how these networks activate in this task and a range of other tasks. So for those of you familiar with looking at brain activity, this is probably recognizable as the response network. We have, this is a right-handed response. So we have left lateralized sensory motor areas here. We have a right lateralized cerebellar activity. So this is a good candidate for a network involved in response. But, you know, that's like interpreting resting state by looking just at the pattern and trying to come up with an interpretation based on the anatomical depiction. But this is the hemodynamic response associated with this network. And I didn't have time to explain exactly how this is computed, but it's not actually taking the bold signal and averaging over trials from these areas. It's a more precise sort of measurement that I don't have time to explain, but it is directly linked. It is basically, these are component loadings. This is basically the component scores. It's actually how the component scores correlate with the finite impulse response model. But anyway, what we have, we can look at the difference between 4 and 6 letters, almost nothing. And we can look at the difference between no delay, which is solid lines, and a delay, which is dotted lines. So there should be 4 seconds between the no delay period and the delay period peaks, because this is a 4-second period of time. And in fact, that's exactly what we find. So when there's no delay, the response of this network is at 12 seconds, but when there is a delay, it's at 16 seconds. And so this fits with what we would expect for a response network. Not really any or much difference between 4 and 6 letters and a 4-second gap between the peak of the two conditions, the maintenance, or sorry, the 4-second and 0-second maintenance period. So, and we also, so if we look at when there is a 4-second maintaining period, we can see that this network peaks at 16 seconds. This one peaks at, we're looking at the dotted lines, 8 seconds. This one peaks at 14 seconds, and this is at 14 seconds as well. So we have peaking at 8 seconds, and then we have 2 peaking at 14 seconds and 1 peaking at 16 seconds. And we know that this is the response network. So we have something early and 2 things sort of in between the response and whatever happened early. So now we start to, you know, think about these phases: encoding phase, maintenance phase, recall. So this is involved probably in recall/response. These 2 are happening in between, and here's what is the earliest type of network. So that's the sort of most basic interpretations. But then if you look at the details of the hemodynamic responses, that gets much more interesting. For example, while the default mode network, these are negative loadings. That's why they're depicted in blue. All the rest are positive loadings. This means that these areas all activate during the task and these ones deactivate. So these are actually the intensity of deactivations. And when there is no delay period, you can see that these solid lines are, you know, coming up to this certain height, and that's deactivation. And there's more deactivation when there's more letters presented on the screen. And then when there is a delay, that deactivation is extended. But the difference between, and so there is load dependence, and that's basically, very, very consistent. The default mode network decreases activity more when the cognitive load increases. That's sort of a standard thing. And the difference, but one thing to notice here is that the difference between 6 letters delay and 4 letters delay. So look, 6 letters, no delay, it's 5 in the morning, 6 letters, no delay, and 6 letters, 4 seconds delay. So the difference in red between the solid line and the dotted line. So there's really not that much difference. So it doesn't really matter that much whether there's a delay period in terms of the overall activity or bold signal measured by this network. So the decreases are around the same, whether there's a delay or not. But if we look at this network, which has also a peak between the encoding period or the initiation and the response, there's really a big difference between when there is no delay and when there's a 4-second delay. And so the difference between this and this when there's a 4-second delay and 6 letters, 6 letters and no delay. And so there's much more difference here. So this, is evidence that this network is actually doing something with maintaining 4 versus 6 letters, whereas this network, not as much evidence for that and more just sort of rote decreases activity for increasing load. And so, let's look at the details of this network here. It's actually a slight delayed response when there is no delay. So these are the solid lines, no delay, and there's an extension of the response like that. But when there is a delay, it kind of drops off quickly. So this seems to be involved mostly because the reason there'd be a delay the reason there'd be an extended response when there's no delay is because basically you're seeing 4 letters and then this extra letter just happens immediately. So it kind of extends out, sort of a visual / encoding type of activity. So we start to come up with some working memory relevant names or that depict the activity that must be involved in each one of these networks. So now I've sort of, by that discussion, gone through quite a few of these different networks already. So there's maintaining, and like I said before, there's many other situations where we see activity in these networks. So the words aren't only from a working memory experiment, but also from a range of other tasks. And this is basically volitional attention to internal representations is what's involved in this maintaining network. Initiation, when there's some delay, it only happens when there is a delay, and then you start doing a task, you see this network activated. So initiating sort of the cognitive processes and not only encoding, but a variety of different tasks. And there's the response networks. I already talked about the default mode networks. There's 2, but there's only actually 1 during resting state. So that I can't talk too much about today. I didn't want to talk too much about auditory perception because that's already quite a standard type of network. So I just wanted to run through 1, 2, 3, 4, 5 different networks and some of the evidence for why we have these words up here for each one of these different networks. So I'll start with extraction of meaning. So extraction of meaning can be linguistic, or it can also be emotional. And when it's linguistic, it's more left lateralized, and when it's it's extracting emotional information, it's actually more bilateral. So this is this is Sam Wong, who did her honors thesis with me at UBC, and now is at McGill in Montreal, and Veena Gagari in Toronto, who we've been collaborating together on analyzing quite a bit of different data. This is her data. So this is a lexical decision task where you just say yes if this is a real word and no if this is not a real word. So this is not a real word, so you say yes and then no. So that's pretty straightforward, but there's a manipulation of difficulty. And so when you're saying, when you have a regular word here, this is just that it is the answer is yes, that's a real word. And it's just a regular type of word, but you can also have a hard condition for saying yes, that's a word. And that's when it's an obscure word that you're not used to seeing very often. And so you would expect to get more activity in a language-based network when the word is obscure and more difficult to process than when it's a regular word. So here's a regular word, dotted lines, and the hard condition is the solid lines. And so you do see an increase. And so there's more taxing of the language network when it's a difficult word. And this is also the case with, networks that are sensitive to task difficulty, like the default mode network. You also see greater deactivity in the default mode network when it's a hard word compared to an easy word. But that type of load manipulation is what we already saw in the working memory experiment. So is this network just sensitive to cognitive load, or is there something going that was different, because this is extraction of meaning, in this case linguistic meaning, or word non-word type of meaning. And so here is a regular non-word. So this is when you say no. So the hard non-word means that it's a set of letters that looks a lot like a real word, but it's actually not. And so in this situation, in order to carry out that task and to respond no to something that looks like a real word, you have to kind of, some type of cognitive process involved in suppressing the response that would be incorrect. It's like, yes, that's a real word, so you don't want to do that. And so you actually see a decrease in the bold signal in the hard non-word condition. So when it looks like a real word, but it's not, your cognitive system does some type of suppressing of this particular network, but not others. For example, the default mode network will be decreased in the hard non-word condition, just like it is in the hard word condition. But in this particular network, which does involve suppression of linguistic information, you see the lowest activity actually in this network, in the language network. So that's a little bit about the extraction of meaning network. How about the reevaluation network? So this is Katie Levine, who finished her PhD with me a few years ago, and Mahesh Menon, who's worked with me. I think Eva probably worked with Mahesh, or certainly knows Mahesh. We're all around the same time, right? Maybe 2010, I'm not sure when, Eva, you were in Canada, but I think Mahesh was in the lab at the same time. But anyway, Mahesh still is at UBC, and we work together all the time. But in this particular task, we're working on having people integrate evidence, particularly disconfirming evidence. This is because we study delusions, which are fixed false beliefs, and integrating evidence against your belief is what we're trying to measure. So we have people rate this. Is this an umbrella or not? Yes, looks like an umbrella. But then we fill in some details in the picture. So this looked like an umbrella, but now when we give more details, you might change your mind. Actually, no, that's not an umbrella. And then you see, yes, it is in fact a bat. So we published a couple of papers on this task, or related tasks. Actually, we published in a related type of task about 20 papers, but because behaviorally, this type of process does correlate with the severity of delusions. But for our purposes today, there's much more activity in this reevaluation network when the evidence is disconfirming relative to when it's confirming. And that's sort of the most basic interpretation of this network, but there are many other situations where we've seen this network. And what's interesting about it is that the peak actually happens after the response. So we think it has to do with, in this situation, thinking things are one way and then realizing, oh, we have to change that, maybe that's not right. So, activity when you need to disconfirm, there's activation in this network. And other times, like during a Stroop task, you might see, oh, was I supposed to read the color? Was I supposed to name the color or read the word? And going through the instructions and questioning yourself seems to activate this particular network. And so I know I'm not getting much time to explain the anatomical differences between these networks, unfortunately, but I guess I just won't have time to do that. But anyway, here's another network. So that sort of covers the three networks that had to do with internal representation. So maintaining is just focusing attention on those internal representations. And in the case that I just mentioned, it had to do with numbers in a working memory experiment, letters. Extraction of meaning. So you see some you might see or hear some visual or auditory representation, have to extract word, non-word, or emotion or semantic meaning from that. That's extractive meaning. And reevaluating is after response, thinking back, did I do that correctly or not? What else might I have to do here? And those conclusions come from matching these anatomical configurations across multiple experiments, and then interpreting how that particular network compared to others, because there's always at least 3 networks per task, and what was unique about that network and its response. Okay, so let's go towards visual. So there's Riley Zurin, who's now moved to Toronto, but he was at UBC working with me, and here's Vina, and this is again her data for Raven's standard progressive matrices. So for this type of task, you have to find what the next response is in this pattern. So 1 square, 2 squares, 1 circle, 2 circles. So if you select 2 circles, get that correct. But if you select, in this case, 3 triangles, 3 diamonds, 1, 2, what comes here, it's going to be the circle, but someone, in this case, chose triangle, so they get that wrong. So there's 3 different levels of difficulty for this task, easy, medium, hard. And when we look at the easy condition, so it's peaking, fairly early and then just dropping off quickly. So in that situation, maybe these 2 look to me to be fairly easy. So there's sort of the answer is quite obvious, and so the efforts put in, it's sort of the attentional resources needed are high and then drops off quickly. When there's a medium, the resources put in immediately are higher, and then it drops off, but maybe not as quickly. So people may still be sort of putting effort in to solving the problem, on average a little bit later. But the hard condition, you might expect it to increase right up to here, which is what we expected, but we found that it actually was down, was lower. So now that we've had so much experience with the human responses, when things, we have to trust that this is actually a good depiction of what's happening in this network. And so why would the hard be lower? Well, it's not lower here, so there's more sustained attention. So it just may be at the beginning that when people, when things it's a difficult problem, people just might have absolutely no idea how to begin and sort of be lost, and just slowly over time keep their attention sustained so that it's a longer period of time. Or it just may be that some people are fast, some people are slow, and so when you average over all the subjects, then you kind of end up with a flatter type of response. And we will continue to investigate that, but that's one example of how we might interpret the sustained attention network. And I know I don't have that much time left, and no one's interrupted me. And feel free to do so, but there's 2 networks left. Now, these 2 networks are actually the latest ones to discover, because all of the other networks that I showed you always activate or deactivate during a task. But these ones, we actually would not publish them because we thought they were error. Didn't really recognize the patterns, and fMRI can pick up movement, and fMRI-CPCA also does sometimes, but the movement is really obvious by being kind of, sort of picking up correlated signal that's kind of just around the periphery of the brain and maybe ventricles. So that's obviously not, that's just an error type of thing. But these ones activate under some conditions and deactivate in other conditions. So it was really hard to kind of, recognize that, because sometimes it'd be in the negative loading, sometimes in the positive loadings. And so, but now we can see that under certain conditions, we see activity, and others we see deactivity. So focus on visual features. This is an early peaking type of network. And so focus on visual features. Obviously, this is, I already probably showed, this is one of the visual networks. And so here's the lexical decision task that we already talked about. And here's the Ravens, task that I already mentioned before. So during the Ravens task, so this there's no difference in this experiment between bipolar and controls, but so you can ignore the difference between the blue and the red line. But there's a early activity in this network and an increase when it's this Ravens type of task. And so that's because the visual details of this task are very, very important. And so you have to, make sure you can tell the difference between squares and circles here. So that's why there's activity. But now we've simultaneously analyzed these, which I don't have time to really explain. But basically, that one network, focus on visual features, was fit to both of these experiments. And it's not fit in the sense that we imposed it. It's just that both experiments were allowed. The restriction was that any networks that were retrieved, we have to see how the activation is in both experiments. So it's exact same network. So you can't sort of compare networks and compare hemodynamic responses. It's better to fix the network and look at the differences in the hemodynamic responses. So, by exploring, we did retrieve this focus on visual features, and there's a decrease during lexical decision. And the reason for that, we think, is that the actual font and visual details don't matter. It can be capital letters, it can be red, blue, italics. It doesn't really matter what the font is. What matters is whether or not this is a word, which is a tasks that doesn't involve visual features. So you actually see a suppression in this network under those conditions and many other conditions. So this will be there to focus on visual features, but when that is not needed, or even when it's required to ignore certain aspects of visual aspects of the stimulus, this will decrease activity. So, and the last network I'll talk about is auditory attention for response. So this is Katie again and Mahesh. We published a paper a few years ago. This is using data from the fBurn consortium, which is an American consortium that has collected 6 different sites in the United States on people with schizophrenia and healthy controls. And this is just an oddball task, so that sometimes there's a higher tone. So this would be, the red is a little bit higher, so people would have to press a button when they hear the red tone. And there was a response in this auditory attention for response network, during this task. And there was this type of suppression for control, so that after responding, this network was suppressed, was not there for patient. I won't talk about that very much. And in fact, we don't completely understand why that's the case, but it does replicate. And so this is, again, auditory attention for response, but this is a different experiment. This is a working memory experiment. And these data were collected. This is, data that you can download from sites that make this available. So these were collected in what we call the LA database. And in this case, there's a spatial working memory experiment, so that people would encode these 1, 3, 5, or 7 dots, maintain for 1, 3, 4.5 seconds, and then there'd be a retrieval. So you have to say whether or not that was a location that was previously presented. And so on the left here is the auditory attention for response network, and on the right is just a typical response network. So this one, you're actually monitoring. This is hypothesized to be involved in monitoring auditory information for response. And this one is basically just pressing the button and responding. And so we see the nice staggered peaks that we saw for the response network in the Sternberg task, because there's a short delay, medium delay, and a long delay. So the response is either, earlier, slightly later, all separated by, 1.5 second delay period here. This actually extends out a bit, but there's the staggered response. So this auditory for response network also has staggered type of peaks for deactivation, but they happen much before the response network activation. So for 1.5 second delay, the peak for response is at 9 seconds, but it's at this decrease is at 7 seconds. So this is analogous, we think, to the type of situation when you need to focus on visual information, just like you do here, that you actually suppress this auditory attention for response network, because you're blocking out, any interfering information in the auditory modality. So, like when you want to talk to somebody and they're looking at their phone, then they don't really hear you, and this kind of is some evidence for that type of cognitive process going on. So, I'll try to leave some time for discussion, but I do have a few slides left, but I'll just go through them very quickly. Anyway, these are the networks that I've already mentioned. And so, again, I think that we have to be careful when we're talking about fMRI and the bold signal, which we're measuring. I don't think it appears to me that it doesn't necessarily map onto the same modular cognitive functions, which are the targets of cognitive psychology and neuropsychology. But it's more of these emergent integrated cognitive processes. At least those are the dominant ones that are detectable by fMRI. And also, interestingly, these cognitive processes are not really accessible by other methodologies. So they're not obviously accessible by structural MRI, no bold signal, EEG or MEG. They're measuring real-time neural activity and not bold signal, so they probably wouldn't pick up these saved cognitive processes. And obviously in resting state, there's no task timing, so you wouldn't be able to pick these cognitive processes up either. I won't go through this. This is just a summary of everything I already mentioned, but in case you didn't pick it up, there's kind of 5 different categories of cognitive processes: attending to internal representations, attending to visually presented stimuli, attending to auditory presented stimuli, and each one of them has a subset, and the response networks, and then these default mode networks. So we've answered these 3 questions. The anatomical characterizations are now classifiable automatically. Cognitive functions, that's obviously what we spend a lot of time on, and these words that we use for labels are always being adjusted. We know that they're not task specific. They're these dominant networks are actually retrievable from all tasks. And so then we get into brain disorder. So I'm out of time. I won't have time to talk about that. But just quickly, we can do things like this. This is people that have been hallucinating in the past week. And in this case, they're listening to speech. And the auditory perception network is hyperactive in people that are hallucinating relative to people with schizophrenia who are not hallucinating, and healthy controls. And that's Chantel's honors thesis, but it's been published, not that particular graph, but that evidence has been published by our group a few different times. Here's the delusions. There's two networks that are involved in delusions, and they were both less active in delusional patients than other networks, but compared to non-delusional patients. But we found that in different tasks. So it depends on not only the network, but also on the task. So this is a probabilistic reasoning task and reevaluation task that I showed you before with the pictures and picture completion. Anyway, so as I already mentioned, it's kind of interesting to be able to measure these cognitive processes, but we're still looking at individual differences and how reliable those are in terms of using this as a way to measure cognition. And then, of course, in the future, we have this network information, and it might be useful to use it with TMS or TDCS or TACS, and you can change the probability or actually activate or deactivate the brain using magnetic or electrical impulses. So I think that this will be definitely an avenue for exploration for the next 20 years or so to see what the limits are. So we do need to start with an understanding of networks, and just stimulating one brain area at a time, probably will be something that, will be in the past one day and not too distant future. So they're the lab members and the collaborators that worked on this, and I guess we have 8 minutes left for questions. And I'm not sure, Eva, if I should stop screen sharing or not.
Unmask yourself. Oh, look, you get a little applause. Thank you, Todd. You can choose if you would like to reappear. Oh, someone's just mowing the lawn outside. Thank you. Thank you so much. It was really nice.
Am I back on now?
Yep, everyone's here. We can see your initials, not your.
So what should I do here?
Right. I'm glad that you've been doing some work in the intervening time that I haven't been there. I'm going to open the floor up to questions. If anyone has a question, just use the little hand doodad. Anastasia, I saw you first. Go.
Thank you. Todd, thank you very much for your talk. Thank you for staying awake for us, or being heavily caffeinated, or both.
Yeah, I am now.
Really, really interesting work. I just wanted a clarification question very quickly. I apologize for the drilling outside my window. So the method of analysis assumes that these signals are time invariant, is that right? We're still compressing them, sort of averaging them out. We are assuming that the same trial will have the same kind of response across time.
Yeah, you're averaging over trials, yes.
Yeah, so we can't capture any slower dynamics, things that would change the hemodynamic response shape over time. So modulatory influences, anything like that, is beyond the scope of the method. Would that be fair?
Well, I mean, you can obviously make different timing vectors for an early, medium, or late stage of the experiment. So to the extent, let's say that you have 50 trials, you could split them into the first 5, second 5, third 5, fourth 5. So you could average over just smaller number of trials and then watch the progression of the hemodynamic response shape over, however long the experiment is. Or you could try to you could code each trial separately and actually have a hemodynamic response for each trial. So that's not normally the way we do it, because we've been sort of trying to get the strongest signal by averaging over more trials, but it's not necessarily, it's not part of it's just how you want to set up the model. You can set the model to be trial specific if you want to.
Okay, great, thank you. That's very helpful. The other question I had very quickly, if I may, Eva, is so how do we think of the underlying physiology when we think of these networks? So my understanding would be that I would have to have a specific question about the specific neurofunctional domain. And if in, oh, there you are, hello.
Hi.
And
Just realized that.
So if I had a task, so say I was interested in auditory hallucinations, for example, like you've shown, right? So if I had a task that had some auditory component and I could activate that network reliably,
Yeah.
if I had any task that could do that, it wouldn't matter what task it is, as long as my question is, do my patients and my controls have a difference in that network, right? So so should we think of the application more in terms of neurofunctional localization, if you'll excuse the term, rather than kind of, where in the brain does this happen kind of approach, if you want to consider the philosophical approach that you might take?
Well, I'm not actually totally understanding your question, but I mean, we do have to keep in mind that we're talking about the correlation between how bold signal is moving. So it's in terms of physiology, we can't really go to the level of neurons. We have to go to the level of bold signal moving around the brain at the same time. So I'm not sure if that is even part of the answer, but maybe you can rephrase the question.
No, that's fair. I guess, okay, I'll clarify my motivation and then hopefully you may understand what I'm trying to say. My motivation is that, for example, if I was interested in the difference between a patient group and healthy controls, I might worry about, whether their gray matter is the same, whether their cerebrovascular reactivity is the same. I don't know if Gabs is in the audience, but she might ask the same question. And so, if you have fundamental physiological questions, does this technique assume that those are equivalent and then looks at neurofunction or correlates?
I mean, sort of. You have to, like I said before, you have to kind of fix one thing. You have to either fix the networks that you're retrieving, or the hemodynamic response can differ. So the way we do a group comparison is that we fit a set of networks to both groups. Okay. And so then we look at the hemodynamic responses that differ between the groups. So you might say, well, this isn't good because one group has, a bunch of gray matter problems, and that may be the case. Like I have a paper with Laura Boyd published on stroke. And in that case, we fit one network to both samples, so a group of healthy people and the people with strokes. But there was just a completely flat hemodynamic response shape in the stroke group. So that meant that they just did not fit the same pattern that the controls fit. Okay, but with schizophrenia, we've done hundreds of studies. We always find, 4 or 5 networks, and quite often, like you saw with the bipolar patient, there's no difference between the networks. So if some of the networks are intact, it can't just be a general kind of, gray matter problem or a neurovascular problem or something like that. And most of the time, the hemodynamic responses match perfectly between the groups. But once in a while, if you split on hallucinations, split on delusions, you'll see that one group on average has a different type of response on one of the networks. And this is not, on one task. So that's sort of the, the way we get around that, concern about, physiological differences between the groups. But we definitely would, not be focused on that. We're kind of making some assumptions, which are checked by looking at the hemodynamic response shape, that there are commonalities between the groups.
That's very helpful. Thanks so much.
Welcome.
Yes, go, Corinne.
Thanks. Sometimes it takes a while to switch a camera on. Thanks for your talk, Todd. And again, I'm just adding my thanks also for getting up at 5 a.m. I wouldn't have done.
Really, no problem.
So what I understand also now during the discussion, and I've played with CPCA a while back, and your talk really, the highlights that I have to return to it. You're constraining in time, right? But also with the number of with the component, the spatial component that you load in.
Well, no, I just want to say that we're not constraining that. We're not loading anything in. Those are emerging in an exploratory way. So there's no constraints, but after the fact, we allow the networks that are there in the data to be extracted, and afterwards we'll take it and we'll compare it to our templates, and then we'll be able to classify it or not as one of the networks that are already known. So there's no constraints on the space. The only constraints are on time.
Just in time. Oh, that's a good clarification. But one question that I still have is also, given your first slide where you say the task-related activity in the bold response is about 10% of the variance, and then there's 90% of the variance unexplained. And of the 90% of the variance, of course, a lot of it is also noise. How does your analysis deal with those noise components?
Well, we put it all into the error. It's all in the 90%, so we don't really pick it up at all. And I mean, there's been some pre-processing that tries to account for, you know, slow frequencies and things like that, but the noise, basically, luckily, using this method, unless the noise is correlated with the timing of the task, then you won't pick it up. And so sometimes movement is a problem. Like we did we analyzed some data that was on pain. So people had pain, you know, applied to their arm, and whenever that happens, people, you know, put their head back because it hurts, right? So there ended up being a movement artifact that was the response of it looked just like a hemodynamic response shape, but it was sort of a, you know, anatomically, it was just sort of, you know, movement. It was nothing. It was just noise, right? So very, very rarely though that happens. Almost always, it's all clean. The components you get out of the task predictable variance.
Yeah, so that indeed, it's a matter of seeing that your design is unconfounded with those forces of motion. That's interesting. Hadn't thought of it, because indeed your reevaluation component that you showed the spatial map for, I mean, it had a lot of edges that were included. And if you're looking at any ICA maps, then often you think like, oh my goodness, to what extent does that component also include noise?
Yeah, that one is, we have to look a little bit more closely at that. I did notice that too. And I don't think that's noise, but I have to check that. There is that, from that perspective, you can see that, yeah.
All right, thank you so much for it.
Thank you, thank you. Does anyone else have any questions? I realize we're sort of out of time, but if there's still hangers-oners who don't have to go to another meeting or whatever, feel free to ask away, unmute, reveal yourselves. Oh, hey.
A quick request. Given that we have now recorded your talk, and I hope you're okay with that, would
I'm
you be
happy with that.
at some other stage return to a neuromethods meeting or something like that, where we can really talk about the nuts and the bolts of the analysis? Would that be something you'd be happy to do at some stage? And I'm putting you on the spot here, so all you can say is yes.
I kind of missed it. Did you say another presentation with more detail about methodology? Is that what you asked?
Well, we have of the presentation, so then it will be more of a discussion, like a Q&A or something like that at some later point, and also at a later time in the day.
I'd love to.
Good. Well,
yeah,
we'll try to set that up. Thanks.
Oh, that's great.
Go to another meeting. So thanks a lot.
I did actually give him that option, and he took this one. So thanks, Corinne. Anyone else? There's people sort of dropping off, but I guess can I ask a question then?
Yeah,
does anyone else have any questions? I guess it was sort of how far I guess the question is like, what direction do you want to go in? Because to me it seems like you're at a bit of a crossroads where you've got this reverse inference kind of possibility where you could go further down, like let's try and explain cognition, or you could go down the pathway of the more sort of treatment-led, you know, basically looking at patient groups and using that information towards treatment. Do you think, where do you see this going, basically?
Well, the easiest, pardon me?
If that's even correct, what I've just, you know, sort of said for you.
Well, you know, I guess there's the easiest problem right now is to try to correlate the activity, what we measure in the hemodynamic response shape with different types of behavior. You know, so what we're doing right now is, you know, the way we sort of identified these networks is by going through the, you know, 200 or so different data sets that are on our own drives and downloaded from international sites. And, you know, we're able to see the consistencies in the anatomical pattern. So then we, you know, compare the way the activation changes over all the different experiments, and then came up with the reverse inference, or with the, you know, description of the cognition. So now, you know, the big question, like we've talked about many times, is always how does the, you know, activity of brain imaging correlate with behavior? So now we finally have developed some multivariate methods that allow us to explore that too. And that's going to be published in Cortex in a couple of months, I think, in a few months. But anyway, So now we're doing that. We're going through all, like, you can download a whole bunch of behavioral data from these online sites, and we're checking to see all the network activity we're using, because the way that, the association between the activity in those networks and the different cognitive tasks changes depending on what cognitive tasks you're trying to predict. So we're running through all the different tasks and symptoms, and we're going to try to get an idea for which networks under which task conditions correlate with, say, IQ, and with, delusions, or with, personality, and that kind of thing. So that's easily doable in a couple of years probably. But then, what we've been talking about is, really, the long-term goal of all of this is to understand the healthy brain well enough to be able to change behavior by stimulating in the right way. And I think a network-based, approach is going to be the future of neuromodulation or neurostimulation. So, but maybe fMRI and looking at bold signal networks isn't the best way, but we'll have to see. So, we're talking about doing that with TMS and sort of testing the limits of connectivity based on, using TMS to stimulate a node in the network. But EEG and MEG, real-time neural activity, I would like to use the same approach we use with fMRI to try to find the networks that are, based on real-time neural activity and not on the bold signal, because that will probably be a better avenue for neuromodulation, at least using, electrical stimulation. You'd want to measure electrically derived networks, maybe different frequencies, different locations, different combinations, and that, is a more longer term plan. So when I have students come in, I suggest they go that way, but I think fMRI is sort of much more, it's just much more established and, more reliable at this point.
Yeah, I mean, for sure, I'd say there's like pros, and cons, as with every technique, but I definitely think the network-based approach to, electroceuticals is a big one. There's a lot of work being done using trying to deliver neurostimulation via a pathway. So since you can't reach deeper areas like hippocampus or amygdala with brain stimulation, they're looking at networks to try and get to it. So for example, there's a couple of parietal sites that you can zap it with TMS and you'll find modulations in hippocampus. And similarly for you can reach amygdala as well through a cortical target that I now forget.
But notice that we're not talking about which task you need to have people do to make these connections.
Yes, these are all resting state based, correct. And yes, I completely agree with you that resting state is a yawn fest. I'm allowed to say that. Any other questions out there in the world? Oh, there's a question in the chat. One simple question: how does one convince a patient with hallucinations to go into the scanner? I don't know.
Well, that's surprising. And when I first started doing schizophrenia research, I was worried about the same thing. But we've seen hundreds, and I'm a part of the International Consortium on Hallucinations research, and they're just people with delusions and hallucinations. They don't, it's only their own internal world that they find disturbing, and the external world is often something they're just as, if somebody else on the ward has delusions, they'll see the falsity of that other person's delusions, and they won't be bothered by it. But, so when you bring the person into this situation, they're just completely fine with it. And even 3 or 4 hours of testing with quite ill patients, they can just seem to focus on what they need to do, and maybe even as a break from their own internal struggles. So yeah, it's surprisingly not a concern.
Okay, anybody else? I think in which case I am supposed to go to a meeting as well. I'm supposed to be in it right now.
Okay, well, thanks for
You're allowed to go back to bed.
Probably not right now.
Too much like coffee. Thank you so much, Todd. That was really interesting. And
thank you too.
We'll see you again at our neuromethods talk then very soon, and I will email you about that. Fantastic. I think that's at the more friendly hour of maybe like 7 in the morning.
Wow, that'll be luxurious.
I know. Good on you. Cool. Thank you, Todd.
Thank you too. Thanks, everyone.
Bye. Bye.
Thanks a lot.